Bone grafting categories. I think it's important to spend some time talking about the regulatory process and pathway by which these products come to market and become available for your clinical use. I think there's a lot of misunderstandings, and I think talking about the regulatory pathway will help us understand in a minute why there's such enormous variability in the evidence, both clinical and preclinical, available to support each of these choices. At the top of the pyramid, the most challenging regulatory pathway belongs to the products that are drug-device combinations. So the FDA reviews these under a pathway which requires an IDE, investigational device exemption, and a PMA, pre-market approval. The second tier category of products really represents the largest in terms of number of products available in the market, and these are the 510K products. So with the introduction of the Food and Drug Act of 1976, the FDA recognized the large number of products that were currently commercially on the market were effectively grandfathered in. Any new products that were to be brought to market, if you could demonstrate they were substantially equivalent to these grandfathered products, you could file a document called a 510K, and upon successful review, those would be cleared by the FDA and could be brought into commercial use. Now, typically, that only requires an animal study, or in some instances, two rather small animal studies to support those filings. The next category, which are the human cell and tissue products, HCTPs, or minimally manipulated homologous tissue. These category of products really have no pre-market review for safety or efficacy by the FDA, but rather the companies manufacturing them have to follow some published guidelines for the proper processing, procurement, documentation, etc. And so these are typically brought into commercial use without any evidence, in many instances, whether that's animal or human. As we discussed, and largely a factor of the regulatory processes, the ranges of evidence that are required to bring these variety of products to market is enormously varied. Most of the companies commercializing these products really don't want to spend much more money above and beyond what's required to get into the market. Rather, they'd prefer to spend their money on marketing, sales reps, commissions, etc. So once again, at the top of the pyramid, the class 3 products requiring an IDE and PMA, those will always have a prospective randomized level 1 clinical study. In the majority of instances, those will be published upon their completion. So you have very solid clinical evidence on which to base a decision to understand both the safety and the efficacy of these products, as those are the FDA's requirements. In the class 2 or 510K products, the FDA's threshold is really more about safety and less about demonstrated efficacy, although they do require at least one animal study. Today in spine, the FDA's requirements for a 510K are a posterolateral fusion in a rabbit model, but historically many products have gotten approved and used in the market on the basis of something as simple as a drill defect in a rabbit femur. Now, if you look at the labeling on these variety of products, you will see some of them are labeled for extremities and pelvis. That typically uses that drill defect model. Those that say spine most typically use the rabbit model with the posterolateral fusion. And as we had discussed previously in the regulatory pathway, the HCTP or tissue products, allograft tissue, or the homologous stem cell tissue products, those typically have no required evidence prior to their submission to the FDA, really just demonstration of their handling properties such to remain safety, try to remain viral inactivation, etc. So typically there are little to no documented evidence in either animal studies or human clinical experience unless physicians choose to do those on their own outside of the auspices of the companies that are producing and marketing these products. I think the first and most important category to discuss in any good bone grafting talk always is autograft. Now, autograft, and in particular iliac crest cancellous bone autograft, has remained our gold standard for performance for many decades. I think there's very little argument to be made that anything to date has been demonstrated to be better than autograft. And if there were just a magic kidney basin in your operating room full of iliac crest autograft, this entire $1.5 billion industry would have never had any reason to be developed. So why is it that autograft performs as well as it does? I think it's because simply it provides all three of the biological functions needed for bone formation, in that it is osteoconductive, osteoinductive, and osteogenic. While I won't be using those terms much through the rest of the talk, I think it's worth defining them fairly quickly. Osteoconduction is the property of providing a surface which is chemically appropriate such that bone can lay down directly onto its surface. Osteoinduction is the property of providing signaling molecules to turn on the native process for bone formation. And then osteogenesis is the property of providing living cells which themselves do the work of laying down the bone. Most typically, these will be mesenchymal stem cells or daughter cells of the MSC lineage, leading down to the end of that lineage for bone formation, which are osteoblasts, the cells that actually do the work of laying down the bone. So in addition to being our gold standard for performance, Autograft is also safe. There's no donor-host rejection potential because it is the patient's own tissue, and there is no potential for disease transmission, again, by virtue of it being the patient's own tissue. While Autograft has been our gold standard for performance, Allograft has been a second-tier choice for quite some time, with a couple of good reasons. The first of which is Allograft tends to be the least expensive option available in most hospitals. It's also been shown to be clinically efficacious, in some low-demand applications such as adolescent scoliosis, certain trauma applications with a good bone bed, and as an autograft extender where you don't get enough autograft in a particular patient. Now, with those advantages come some significant disadvantages. Because allograft is human tissue, it carries a potential for disease transmission. Now, while the processes for reducing those disease transmission potentials have improved over the course of years, that potential can never be eliminated, can never be brought to be zero. Now, along as we've discussed, the regulatory pathway for allograft tissue is guidance documents. There is no pre-market review for safety and efficacy. Merely the processors must follow guidelines in how they procure the donor tissue, how they process it, how they distribute it, and also the keeping of documents of what patient it goes into, tracing all the way back to the original donor.